This is TANUVAS Global Veterinary Large Animal Science Webinar Series 2020. Yesterday we had a topic on reproductive ultrasound in mass and stallion by renowned theologian Dr. Carla Carlton, Professor Emeritus Large Animal Clinical Sciences, Michigan State University, USA. For those who have missed, it is available in YouTube for following it up. Today we have two topics. The first one is modulating adipose tissue function in periparturient cows by Dr. Andres Contreras. The second topic is on Michigan State University colic in horses for those by Dr. Elizabeth Carr. Good luck for all. May I request Dr. Siju Simon to introduce the first speaker.
Good evening and good morning. It's my privilege to introduce the speakers of today's two-day webinar. And first speaker is Dr. G. Andres Contreras. He is an associate professor in the Large Animal Clinical Science College of Veterinary Medicine, Michigan State University, USA. The topic for today's lecture is modulating adipose tissue functioning in periparturient cows. Andres Contreras received his DVM from Universidad Nacional de Colombia. After 3 years of large animal critical practice in Central Colombiana, he served as intern in the dairy internship program at the College of Veterinary Medicine, Michigan State University. He continued education, received a master's degree in mastitis and Ph.D. in competitive medicine and integrative biology. His research background includes expertise in adipocytes and endothelial biology, as well as in adipose tissue synthetic innovation. His research work has focused on the interactions between adipose tissue function and diseases. His doctoral studies evaluated the effect of lipolysis on endothelial cells' inflammatory response. His postdoctoral research work focused initially on the lipolysis-induced white adipose tissue remodeling processing and developed into the elucidating the effect of sympathetic innovations on adipocyte phenotype and functions within the subcutaneous adipose tissues. With this introduction, I would like to invite Dr. Andrew sir to convey the lecture. Thank you.
Yeah, over to Dr. Andres.
Good morning, good evening. Can you hear me okay? Can you see my presentation okay?
Yeah, it is okay, perfect.
Okay, good. So, well, first of all, thank you very much for the invitation. It's an honor for me to speak in your seminar series. And let's get into my presentation. So today I'm going to talk about how we evaluate and modulate the adipose tissue function in periparturient dairy cows. But let's first take a look at the overview of my presentation today. First, I'm going to talk about and define lipid mobilization. Then we'll start to get some background on the impact of lipid mobilization in the adipose tissue, and that's usually defined as adipose tissue remodeling. They will look into what happens to the adipose tissue function in common diseases that include endotoxemia, and they will touch upon ways in which we could modulate adipose tissue function, and then we'll conclude with conclusions and future directions. So let's start by defining the term lipid mobilization. It is composed of two processes. One is lipogenesis, and the second one is lipolysis. I'm pretty sure you're all aware that lipolysis is the breakdown of the triglyceride molecule within adipocytes and many other cells that do have lipid droplets. And lipogenesis is the process by which fatty acids or other energy sources are assembled into the triglyceride molecule within lipid droplets. Usually can be adipocytes, but other cells such as myocytes and hepatocytes can also store triglycerides as lipid droplets within lipid droplets. What happens to the periparturient period? Are assembled into the triglyceride. Dairy cows and many other species enter a phase of negative energy balance. That's usually because cows have a very low dry matter intake, run around calving, and there are many endocrinological factors that actually promote the funneling of energy towards the mammary gland, and this causes negative energy balance. During the periparturient period, that negative energy balance is countered, or is reduced by increasing lipolysis. Why? Because lipolysis releases free fatty acids that can be used as energy source in many tissues, including the liver, the muscle, the heart, and also can be channeled through the mammary gland to provide energy to the neonate as the fat in the milk. So during the periparturient period, as I said before, there are certain endocrinological signals that actually promote lipolysis. One is that cows and many other mammals develop a state of transient insulin resistance. As you know, insulin is the major anti-lipolytic hormone. So when insulin is not sensed by the adipocyte, lipolysis is dramatically increased. Also, there are at least two hormones that are increased during the periparturient period. One is the growth hormone, and two, catecholamines released by the sympathetic nervous system. So those two promote lipolysis. So as a consequence, during the periparturient period, you have a high flow of free fatty acids into blood, and that causes many consequences. The one and most obvious is body weight loss. On average, a dairy cow loses between 5 to 10% of her body weight within the first 30 days after parturition. And this is from a very nice study from Israel that they actually assess the body weight loss in cows. So they collected data from about 2,200 cows from several large dairy herds in Israel. And they weighted those cows daily for about 120 days. And as you can see here, as you can see here, the body weight loss is concentrated within the first 30 days after parturition. Here on the top squares, you have the body weight loss of first lactation heifers. The second set of the squares here is the body weight loss of second lactation cows, and the lower is the body weight loss of third or more lactation cows. And as I said before, all the body weight loss is concentrated within the 30 first days after calving. So what happens to the adipose tissue during that body weight loss of 5 to 10%? It is very difficult to measure adipose tissue depot mass loss because that involves euthanizing the cows and actually weighing the fatty acid, the fat depots of the cow to be able to assess that. And even with that, you need several animals because it's very difficult to follow one single cow through the transition period into lactation. However, a group of scientists in Germany, they were able to do this kind of research, and actually they collected samples from a group of 20 cows, and they euthanized those cows, separated in 5 groups, obviously, at 1, 42, and 105 days after calving. And what they did is they weighted the different adipose tissue depots. And I want you to focus on the weights at days 1 and 42, which are the ones that reflect the body weight loss during the periparturient period. And as you can see here, just basic arithmetic calculations, those cows between day 1 and day 42 lost about 12% of their carcass weight and about 24% of the weight of the subcutaneous depot and about 34% of their visceral depots. So the body weight loss that dairy cows experience during lactation is mainly due to losses in the adipose tissue mass. Okay, so now let's look at how is this lipolysis in other words, lipid mobilization regulated in periparturient cows. As I said before, there are two major players at the endocrine level. One is insulin and neuropeptide Y, which are reduced and therefore promote lipolysis. But also, there are two hormones, the growth hormone and the natriuretic peptide, and also the sympathetic nervous system activation that actually promote lipolysis. At the paracrine and autocrine level, there are different factors that promote lipolysis and inhibit lipogenesis around parturition. One is the release of specific cytokines and adipokines that promote lipolysis, and those are the inflammatory cytokines such as IL-6. And the second thing is the release of lipolytic products such as fatty acids and oxidized fatty acids that actually promote lipolysis because they influence mitochondrial respiration. When mitochondrial is not working appropriately, it induces the release of reactive oxygen species that promote lipolysis. So around calving, the cow is in a pro-lipolytic environment, and that's why free fatty acids are released at high concentrations in plasma. So what happens to the adipocyte? That's the key cell within the adipose tissue that stores the triglycerides during the periparturient period. So here we have an adipocyte, and you might think that what happens during lipolysis is that the adipocyte lipid droplet just shrinks, is reduced in volume. But that is not the case. What happens during lipolysis is that the lipid droplet starts to get fractured into different smaller lipid droplets. And what's really interesting is that those lipid droplets are targeted to the organelles that are able to use the fatty acids released by lipolysis into different functions. So some lipid droplets are targeted to the RER, the reticulum endoplasmic, RAF endoplasmic reticulum, and some other ones are targeted towards the mitochondria. Some additional ones are targeted near the nuclei, and there they can activate different programs within the nuclei that are able to facilitate the use of free fatty acid as energy. Within the endoplasmic reticulum, free fatty acids can be targeted for export when they are packed with fatty acid transporters, or they can be funneled back to a lipid droplet to be resterified into triglycerides. And this is an electron microscope picture showing how is that the lipid droplets are fractured, as you can see here, and how they are targeted to either the mitochondria or the endoplasmic reticulum here. And this immunofluorescence microscope images show lipid droplets on green and mitochondria on red. And as you can see, as lipolysis increases in rate, all the lipid droplets are targeted towards the mitochondria. So during the periparturient period, lipid droplets are targeted to organelles that can use fatty acids. However, when lipolysis is very intense, the lipid mobilization function within adipocytes gets affected, and we call that state lipolysis dysregulation. So what happens during lipolysis dysregulation? Excessive release of free fatty acids activate the inflammatory responses in the nuclei, especially through the activation of NF-kappa B, other cytokines, and the recruiter of macrophages that is called osteopontin. At the same time, because a lot of fatty acids are targeted to the mitochondria starts to release reactive oxygen species that further activate different enzymes that can induce the release of lipid mediators of inflammation, such as prostaglandins and thromboxanes. So excessive lipolysis leads to inflammation within the adipose tissue. And here you can see how activating lipolysis excessively can lead to fragmentations of lipid droplets within the adipocyte, And this picture is from a study that used a specific mitochondrial dye that glooms red when it's activated in the mitochondria. So as you can see, when you activate lipolysis with the beta-adrenergic agonist CL6, mitochondria are greatly activated, and this coincides with the fragmentations of lipid droplets. So now that we saw what happens at the adipose tissue level with shrinking adipose tissue depots, and at the adipocyte level with fragmentations of lipid droplet and inductions of inflammation within the adipocyte, now let's look at what happens to a cow. Let's go back to the cows. So when there is excessive lipid mobilization, dairy cows have increased susceptibility to diseases. And this has been well studied all around the world. There are studies from North America and Canada, studies from Germany and other regions of Europe, and studies in South America and Europe too. And what they show is when cows have high rates of lipid mobilization, and that is reflected in high concentrations of free fatty acids in plasma and beta-hydroxybutyrate, cows are at a higher risk of having periparturient-related diseases. And what's making it worse as we conduct genetic improvement in our cows is that we are selecting for cows that actually have a higher lipolytic response. As shown recently by Dr. McNamara at Washington State University, that he shows that cows that have high genetic METIC are more prone to have excessive lipolysis. So what are the consequences of excessive lipid mobilization? First, there is an increased risk for having diseases, actually about 1.5 times compared to cows that have low lipid mobilization rates. And this is especially important in cows that show levels of free fatty acids that are about above 0.3 milliequivalents per liter 3 weeks before parturition, or above 6 milliequivalents per liter 6 weeks, between 1 and 6 weeks after calving. This is also true for cows that have BHB contents above 10 milligrams per deciliter in blood. Cows with those two elevated markers post-calving have higher risk for retained placenta, metritis, displaced abomasum, and mastitis. And they also have higher rate of clinical hyperketonemia, which is a comorbidity in nearly 60% of the clinical diseases in periparturient dairy cows. Why excessive lipolysis induces or increases susceptibility to diseases? The response is because free fatty acids affect immune function. For example, high concentrations of saturated fatty acids reduce phagocytosis efficiency and cell viability in polymorphonuclear cells and macrophages. Also, they do increase the secretion of pro-inflammatory cytokines and reduce the proliferation and Ig secretion of B cells, and they reduce the activation and polarization of T cells and natural killer cells. All this is accompanied by impaired chemotaxis. So what you have in a periparturient cow that has excessive lipid mobilization is dysregulated inflammatory response that have very low phagocytosis efficiency, impaired chemotaxis, and very intense secretion of pro-inflammatory cytokines. So they enter a state of low-grade inflammation right after calving. So as I said, excessive lipolysis leads to dysfunctional inflammatory responses, and they are characterized, as I said before, by delayed and impaired phagocytosis activity, inefficient pathogen clearance, and that's because phagocytosis is altered, excessive pro-inflammatory cytokine secretion, especially TNF-alpha and IL-6, extended resolution of the low-grade inflammatory process, and what's most important is the adipose tissue is one of the key organs where these inflammatory processes occur in the cow. At the same time, excessive lipid mobilization has a negative impact on lactation performance and reproductive efficiency. Cows that have high NIFA and high BHB after calving, they have milk yield reduction of about 600 kilograms per lactation. And the pregnancy risk of those cows is nearly 20% less than in those cows that have low rates of lipolysis. So up to here, I hope that I have give you a good background of how lipid mobilization occurs during the periparturient period, what are the effects on adipose tissue, and how is that it impacts immune function and milk production in dairy cows. So now let's look at what happens within the adipose tissue in the process of adipose tissue remodeling. As I said before, there is a reduction in loss that is largely explained by reduction in the adipose tissue mass, and this is mainly explained by the reduction in the adipocyte size distribution. So this image, this figure, is from a study that we performed in 2017, where we actually measured the adipocytes in adipose tissue from dairy cows at 3 different stages. G1 was about 10 days before calving, G2 was about 5 days before calving, and PP is periparturient, that's about 7 weeks, 7 days after calving. And as you can see, what happens when cows starts to mobilize fat is that the number of adipocytes in the smaller size, this is about 2,000 to 4,000 square microns, is dramatically increased in cows after calving, and the number and distribution of adipocytes in the larger sizes, that's about 8,000, is dramatically decreased. So this shows how the adipocytes is reducing. But at the same time, lipolysis induces a process of adipose tissue remodeling that is characterized by 3 things. One is an inflammatory response that induces immune cell infiltration. The second one, there is adipocyte proliferation because there are some cells that do die and they need to be replenished. And the third one is extracellular matrix remodeling because the adipose tissue needs to accommodate to the smaller size. Our lab is particularly interested in the inflammatory response and immune cell infiltration because of the consequences to animal health and animal welfare in cows. So let's look at what happens in the cows when there is high rates of lipolysis after calving. As you might think of, adipose tissue is mainly composed of adipose tissue, adipocytes. But besides adipocytes, there are other cells within the adipose tissue, and those include macrophages, pericytes, preadipocytes that are the adipocyte progenitors, monocytes, and endothelial cells. What happens during lipolysis is that the macrophages start to sense that the adipocytes are releasing too much fat, and they actually have to contain the lipolysis products that are actually toxic to the cells. So macrophages actually start to engulf lipid remnants and localize them. So these two are pictures from a study that induced lipolysis in rodents. And as you can see here, the lipid droplets are in gray and the macrophages are marked in green. And as you can see, after induction of lipolysis, macrophages that are in green start to internalize lipids to localize the fats that are being released from lipolysis into the adipose tissue milieu. So macrophages are very important to contain the release of free fatty acids into the adipose tissue. And macrophages are not of the same phenotype within the adipose tissue. There are some macrophages that are of the M1 phenotype, and there are other macrophages that are of the M2 phenotype. When there is intense lipolysis around calving, adipose tissue remodeling occurs with infiltration of a lot of macrophage type 1, macrophages of the type 1. What happens with this type of macrophages is that, at least in humans, they have been linked to the development of insulin resistance in adipocytes. And this leads to enhanced lipolysis. And this results in chronic elevation of free fatty acids, at least in humans. But only until recently, we didn't know what type of macrophages were infiltrating the adipose tissue of cows during the periparturient period. And in 2016, and 2015 and 16, we did a series of studies looking at characterizing the adipose tissue macrophage infiltration in cows that were in the periparturient period, and we compared those to cows that were obese, that were overconditioned. So we collected a set of samples from overconditioned cows that were over 300 days after parturition and that had a very high body condition score. And we also collected adipose tissues from transition cows that had displaced abomasum and were very early into lactation, only 7 to 8 days after calving, and that had a very low body condition score. We collected samples from the omental adipose tissue and from the subcutaneous adipose tissue at the moment of correction of the displaced abomasum during surgery. And we performed qPCR, flow cytometry, Western blotting, and immunohistochemistry analysis. So after we collected those adipose tissue samples, we found that in cows that had DA, there was an increased infiltration of macrophages, as you can see in these images. In brown is the macrophages, because we stain with a macrophage-specific marker, in this case CD172A. And as you can see, macrophages are localized surrounding adipocytes that are either dying or are starting to break up and releasing the lipids. And this is an H&E hematoxylin eosin photograph showing you how in detail are the different nuclei surrounding the remnant of an adipocyte. We termed this a crown-like structure or adipose tissue aggregates, and we started to count them in both the omental and subcutaneous adipose depots in cows with DA and in cows that were in an anabolic state or were dry, as I showed you in the previous slide. And what we found is that there was a significant increase in the number of these adipose tissue aggregates per 100 adipocytes in cows with DA in both the omental and subcutaneous depots compared to those cows that were not in an intense process of lipolysis, and that was true for both sites. The next thing that we wanted to do was to analyze what was the type of macrophage or phenotype of macrophage that was infiltrating those adipose tissue in both cows that were in an anabolic state or with DA. And what we found is that, yes, there was an increased number of macrophages infiltrating, and this was shown by the pan-macrophage marker CD172A, and this is using flow cytometry. But what was really interesting is that cows that had DA had a higher number of cells that were expressing CD14 and CD11C. Those two markers are specific of the M1 phenotype. But at the same time, those cows had more macrophages of the M2 phenotype. So in general, lipolysis increases infiltration of macrophages of both phenotypes, M1 and M2, into the adipose tissue in cows. But when cows start to have excessive lipolysis, such as in this case, cows with displaced abomasum, they do have more cows of the M1 type, more macrophages of the M1 phenotype. So we concluded from this study that cows with DA and ketosis have a higher infiltration of adipose tissue macrophages that are predominantly of the M1 phenotype. And this was characterized by cell surface makers CD14 and CD11C, and by a specific gene expression of TNF-alpha, IL-6, and CD14 that are all genotypic markers of M1 phenotype. What was really interesting is that overconditioned cows, in contrast to obese rodents and humans, do not present adipose tissue macrophage infiltration. because there are very few adipose tissue macrophage aggregates. So in cows, high body condition score does not equal inflammation in adipose tissue, in contrast to what's observed in rodents and humans. So this is our working hypothesis of what happens during the periparturient period. So in a normal cow, she will exhibit moderate rates of lipolysis that induce a very moderate infiltration of macrophages that the majority are of the M2 phenotype, and very few are of the M1. As lactation progresses, lipolysis starts to decrease, and cows go into a productive lactation with very high reproductive efficiency. However, when cows experience excessive lipolysis, what occurs is that there is a very rapid and intense infiltration of macrophages, where the majority of those cells are of the M1 phenotype, that actually causes the development of crown-like cell structures or adipose tissue aggregates that actually promote lipolysis and sustain lipolysis into lactation. So these are the cows that are more susceptible to disease and that might have lower milk production and lower reproductive efficiency. So now let's get into how is that we evaluate adipose tissue function and when we should evaluate adipose tissue function in dairy cows. So as for the when, we should actually start to monitor how is the adipose tissue remodeling and the lipolysis intensity occurs through the periparturient period. So we should start evaluating the adipose tissue function early during the dry off, when we dry those cows off, when we stop milking. Then as cows approach parturition, about 2 weeks before calving. Then at fresh, because we're able to evaluate actually the intensity of the adipose tissue lipolysis, and then at peak lactation, because it's a good way of assessing how well we're taking care of the periparturient cows in our farm. How can we measure adipose tissue function? Well, there are many ways, there are many approaches. The most basic ones are body condition scoring at body weight at the different time points that I referred in the previous slide. Also, continuous monitoring of blood markers of lipolysis, which are NIFA and BHB. And for those ones, there are thresholds that are already well described in the literature, and those include herd alarms, where you would know if your cows are at a higher risk of lipolysis dysregulation and therefore increased susceptibility to diseases. Also, you can evaluate milk fat to milk protein ratio. When this ratio is high, that would indicate that cows have a high rate of mobilization of lipids post-calving. And there is another way, which is the use of image biomarkers. Image biomarkers are basically ultrasound images of the adipose tissues in the different locations in the cow. We usually monitor visceral and subcutaneous fat. And what's really interesting is that recently, a group in Germany that I cite here in this paper from 2016, they actually developed models, regression models, that are able to predict the mass of different adipose tissue depots. In this case, we have a convex ultrasound using a convex probe, where you can see the subcutaneous adipose tissue here on top. And what you do is you measure the adipose tissue depot and you enter this reading into the regression models that this group developed, and they're able to actually calculate the adipose tissue mass. The same is for this visceral adipose tissue depot. In this case, actually the fat surrounded the kidney, and the subcutaneous adipose tissue depot, in this case at the rump, where you can measure the subcutaneous adipose at the 12th rib. What's important about these image biomarkers is that they are very specific to the adipose tissue. Unlike BHB and NIFA, they are not. because they can be altered by the diet or, for example, by feeding patterns, the depth of adipose tissue depots cannot be altered by that. So they are very specific to the adipose tissue. And what we found in the study in 2015, is that the subcutaneous adipose tissue depth is strongly associated with NIFA. So we can actually predict the intensity of release of NIFA by measuring the changes in subcutaneous adipose tissue depth between the close-up period, so about 2 weeks before calving, and the reading at about 1 week after calving. There are cons about using image biomarkers, so ultrasounding. First, the cost, especially for smaller herds, also is very labor intensive and needs a person that's knowledgeable of the technique and that can use the ultrasound, but also at the same time it requires convex probes, and those are not very common in pediatrics practice. Also, at this time, there are not enough large epidemiological studies that associate the depth of, especially of visceral adipose depots, with the development of lipolysis dysregulation. I hope in the future we're able to use ultrasound as a tool to evaluate lipolysis dysregulation more heavily, and for that we really need large epidemiological studies that present associations with specific diseases, especially those with high impact to dairy cows, such as displaced abomasum, ketosis, and fatty liver. So now that we have seen what happens with lipid mobilization, adipose tissue remodeling, so now let's look at what happens to the adipose tissue when there is a disease, an inflammatory diseases, especially bacterial diseases. And in the past 3 years, we have been looking at what happens when the adipose tissue is exposed to bacterial endotoxin. We have focused on LPS, which is obviously from gram-negative bacteria, but also there are studies showing that gram-positive cell wall, especially lipoteichoic acid, is able to induce lipolysis. So during lipolysis, LPS, during disease, LPS is actually able to induce lipolysis in adipose tissue by two pathways. One is the classic pathway. Actually, LPS is able to activate the toll-like receptor 4, and through this pathway is able to increase the phosphorylation of hormone-sensitive lipase, and this releases free fatty acids into circulation. And when LPS activates toll-like receptor 4 and induces the activation of NF-kappa B, it also is able to activate lipolysis. So in humans and in rodents, it has been shown that when a person has endotoxemia, the adipose tissue starts to release free fatty acids because the LPS activates these two, the classic and inflammatory pathways of inflammation. So what we didn't know at this time, until we did the following studies that I'm going to present to you, is if this was the case in cattle. And then we found that yes, that is the case. LPS can induce lipolysis in cattle, at least in adipocytes in an explant model. So to assess this, we collected subcutaneous and omental adipose tissue explants, and we exposed these explants to different concentrations of LPS, 0, 1, 10, 20, and 100. And we actually exposed these at the same time that we were activating lipolysis using a beta-adrenergic agonist isoproterenol. And we assessed the rate of lipolysis using measuring the concentrations of glycerol that were released into the media. And as you can see here, LPS is able to increase release of glycerol in adipocytes when they are exposed to LPS for 3 hours. And the rate of release is very similar to what is observed when we activate lipolysis with an adrenergic agonist such as isoproterenol. What's more interesting is that the release is more intense from visceral adipose tissue, in this case, the omental adipose tissue, when we compare that to subcutaneous adipose tissue. So if a cow has endotoxemia, the adipose tissue will increase the release of free fatty acids, especially in the visceral sites. But when we have endotoxemia, the activation of lipolysis is not short-term, because usually endotoxemia lasts several hours. In the previous set of experiments, we exposed the cells for 3 hours to LPS. But in this next set of experiments, we exposed the cells for 3 and 7 hours with different treatments. That included LPS at 20 micrograms per ml, isoproterenol as a positive control for lipolysis, and then insulin to evaluate if LPS actually impair the anti-lipolytic effect of insulin in adipose tissue explants. And what we found is that as we extend the exposure of adipose tissues to LPS, the lipolytic rate actually increases. So it is expected that during the endotoxemia, the lipolytic rate is only going to increase within the first state of endotoxemia as the time goes by, as you can see here, at 3 and 7 hours. And this increase in lipolysis is partly mediated by the reduced sensitivity of adipose tissues to the anti-lipolytic effect of insulin. In this graph, we have the reduction of glycerol release as a response to insulin in the media, and the different treatments that we have that include LPS, isoproterenol as a positive control for lipolysis, and insulin. As you can see, in cows that in explants that have not been exposed to LPS, when you add insulin, the lipolytic responses is greatly reduced. However, when there is exposure to LPS, the explants do not respond to lipolysis. So during endotoxemia, the adipose tissues develop lipolysis dysregulation. That is for several reasons. First, because endotoxin, in this case is LPS, triggers lipolysis. And at the same time, endotoxin impairs adipocyte insulin sensitivity. So this tells us that during endotoxemia, cows will have a predisposition for metabolic diseases and clinical hyperketonemia. So now let's move to the last part of my talk, where we are going to go over a few tools that we have available to modulate adipose tissue function in periparturient cows. The first one is the supplementation of niacin. Niacin is vitamin B3. It's usually referred as nicotinamide or nicotinic acid, but only a nicotinic acid has anti-lipolytic effects. Niacin acts on the adipocyte receptor GPR109, and through the activation of this receptor, what it does is that it inhibits the phosphorylation of HSL, which is the key hormone in lipolysis, and this also has some vasodilatory effects. So niacin can be used as an inhibitor of lipolysis or during heat stress as a countermeasure for heat stress because it induces vasodilation. Niacin is easily degradable in the rumen, so niacin needs to be fed as rumen protected. The doses that are used currently in cows are between 6 to 24 grams per day. But what's really important is that you have to feed niacin before and after calving. If you only feed niacin after calving, there will not be a strong anti-lipolytic effect, and that will not reduce free fatty acids and milk fat after calving. So you can assess the efficiency of your niacin treatments by evaluating free fatty acids after calving and by evaluating the milk fat to milk protein ratio. And those two should be reduced if you feed up to 24 grams per day of niacin before and after calving. A second option that we have available is to include chromium in the diet, especially after calving. There are at least two sources of chromium, chromium propionate and chromium methionine. Chromium picolinate is also available, but it's not as broadly used as chromium propionate. Chromium does not have a direct effect on lipolysis. However, chromium enhances lipogenesis. As you saw in the first slide, lipid mobilization is the balance between lipolysis and lipogenesis. When you have increased lipogenesis, you will reduce lipolysis. Chromium, what it does is that it increases the activity of the insulin receptor through the activation of chromoduline. And this is especially effective when you have diets with high content of corn. A third approach to reduce lipolysis in periparturient cows is the use of saturated and monounsaturated fatty acids as mixture in the diets of fresh cows. There are many reasons why this supplementation will reduce the mobilization of lipids from fat. One is because they induce increased energy intake. And the second one is that when you combine palmitic acid and oleic acid, which are the two fatty acids that are more well studied, you actually get a promotion of lipogenesis. If you feed palmitic acid alone, you get an increase of milk production. However, there is higher lipolysis rates. However, when you start to include an unsaturated fatty acids such as oleic acid, you can maintain the milk production and you reduce lipolysis. And this is because oleic acid promotes the release of insulin. So when you combine both, you get an increase in dry matter intake when you have palmitic acid and oleic acid combined, and sustained production. This is energy corrected milk. And then you reduce the body weight loss, as you can see in the top image. Why is that this happens? I'm going to jump into this slide quickly because I'm running out of time, as I can see. Oleic acid actually promotes insulin sensitivity within the adipocytes. And we did this, we assessed this in using explants in experiments where we expose cows to a controlled diet that didn't have any oleic acid or a diet that had oleic acid. And what we found is that when you start feeding oleic acid in the presence of palmitic acid, the cows have a higher antilipolytic response to insulin when you stimulate lipolysis with isoproterenol. And this is reflected in this slide also when you have a lower release of free fatty acids in the presence of insulin. So as a conclusion, we can say that a successful transition from gestation to lactation requires that adipose tissue efficiently integrates systemic and metabolic and inflammatory signals, and that lipolysis is an inflammatory event that induces remodeling in adipose tissue, and that endotoxin induces a lipolytic and inflammatory response that might impact the adaptation of adipose tissue to a periparturient period. We have available nutritional, such as oleic acid, pharmacological, such as niacin and chromium, and management tools to reduce lipolysis. However, currently, we have a need to increase the number of pro-lipogenic interventions that will minimize the release of free fatty acids into circulation. This will actually reduce the deleterious effects of NIFA without any impact on lactation performance. And this is all what I have for today. So now I'll be happy to take any questions that you might have. Thank you for the opportunity.
Thank you, Dr. Andres. We have enjoyed your lecture. There are many questions, or important questions we are putting to you. During the course of lecture, you told about the usage of corn.
The usage of what?
Usage of
Chromium?
corn. Corn to during explanation of the saturated and unsaturated, high amount of corn, yeah, reduces lipolysis. Would you please elaborate on that?
Chromium or oleic acid?
You told high amount of corn reduces the lipolysis. Could you please explain on that?
Is he referring to chromium or oleic acid? I didn't get the question. Sorry.
So what is the mode of action of this palmitic acid and oleic acid?
Oh, okay. Yeah. So the mode of action for palmitic acid actually is the combination. You have to combine both. So if you use palmitic acid alone, I'm going to share again here the slide very quickly. And you'll be able to see what I'm referring to. So here, when you feed palmitic acid only, you get an increase in milk production. However, palmitic acid only promotes lipolysis. However, when you add oleic acid into the diet in the presence of palmitic acid, you actually maintain milk production, but you reduce lipolysis. And this is through an increase in insulin sensitivity. And these experiments here show that when you have oleic acid in the mixture, you actually reduce the release of glycerol when you stimulate the explants. In this case, this was an ex vivo experiment, the explants to adrenergic stimuli. So the presence of oleic acid reduces lipolysis, as you can see in this slide. And when you include insulin, if you compare the lipolytic response of the controls and the explants that were exposed to oleic acid, actually the explants that were exposed to oleic acid have a higher response to insulin. So the mode of action is through the increase in insulin sensitivity by the adipocyte, and that's driven by oleic acid. The mechanism is not clear yet. There might be two sub-mechanisms. One is oleic acid has been shown, at least in rodents, to increase the production of insulin by the pancreas. And the second one is that oleic acid might promote the reesterification of fatty acids into small lipid droplets. So, as I said, if you have more lipogenesis, you will have more lipolysis. But the mechanisms, exact mechanisms, are not clear. Two hypotheses: increased production of insulin by the pancreas. A second mechanism, increased lipogenesis by enhancing reesterification of fatty acids in the adipocyte. I don't know if that answers your question.
Thank you, Dr. Andres. Then during the course of the lecture, you told chromium, different chromium propionate and like that. So which do you recommend?
The most common is chromium propionate, and there are many studies, especially here from the US, using chromium. However, the problem is that if you start, the problem is that depending on the source, there have been conflicting results. So chromium propionate is the one that has been shown better results. However, we still need to do more research into the mechanisms by which it acts in the adipocyte. It is clear in rodents, but it's not clear in cows. So that's why I didn't cite an exact mechanism.
Okay, is there probable reason that you have not mentioned the dose for the ruminants? Is it?
Yeah, and there are three different formulation sources. So it depends on the type of chromium. I don't know if you have different formulations of chromium available where the person that was asking the question is. But if he contacts me, I'll be happy to go over specific doses depending on the type of chromium that you have available to use.
Thank you. Regarding the niacin supplementation, you have mentioned about rumen protected. So how to do that?
Well, I think we have rumen protected niacin available in several parts of the world.
Yeah.
It's usually formulated to mix with the grain.
Okay.
So yeah, it's available. Rumen protected. There are many, at least 3 companies that I know of that have it available in Asia, Europe, and here in the Americas. So I don't know if you have it available there. I don't think if using it IV might be an option, because you really need to have continuous supplementation of niacin in order to be effective, right? And important that you start the supplementation before parturition. There are studies where they started supplementation only at calving, and it doesn't work. You need to prepare the adipocyte to actually be able to reduce the response. I think I lost my camera here. I don't know if that answers the question.
Yeah, you answered. You got the point. So we learned that it should be given before and after calving.
Yes, niacin important before and after.
So regarding the image biomarker, you said on 12th strip, is it so specific? Why it is recommended at the site? What is the importance
of the lubricant or oil between the probe and that's what you're asking? Or what?
No, during the coverage on ultrasound, image biomarker, the site chosen is 12th rib.
So, yeah, 12 rib. And I cited that site. I'm going to go again to the presentation in a second here. The site that I cited is because that's where, and I really refer you to this paper, because that's where we have a regression model available based on the studies by Rashka et al. In 2016. So if you do it at the 12 rep, you will have a formula to calculate the adipose tissue mass because they validated it in that site. Okay.
Yeah, we already well-calculated regression model is available, so it is easy for us. You mean to say that? Okay.
Yes.
Thank you.
Yes. Okay.
Because
thank you. Sites it's not calculated.
Thank you, Andres. We enjoyed the whole of the class, and it was very informative, and we got more important points on niacin, chromium, and this combination of palmitic and oleic acid. We enjoyed. Thank you so much. Thank you for sparing your valuable time in discussing these points. Definitely, the students and practitioners would have got benefited. Thank you so much.
Thank you.
Thank you.